Thank you very much. It's a great honor to be able to join this symposium. It's been very eye-opening. For me, I'm actually very fortunate to be at a joint position as a pediatric dentist, and I also spent 7 years of training in microbiology. And that helps me to answer a lot of questions my patient always asks me, and so does myself too. And I was very fortunate, happy to be at UCSF, which when I run into roadblocks and I have the saviors from School of Pharmacy and School of Medicine with those real basic scientists, and saved my project for going on. So I'm going to share one of my projects that's actually supported by Can-Do Center 2 times. I'm just going to start with talking about dental caries is an infectious disease. I know Dr. Benzion don't think it is, but for me as a microbiologist, I want to emphasize it is an infectious disease, not so much as like polio or other specific infections. We see it's an infectious disease because especially early childhood caries is characterized with high-level infection of Mutans Streptococci. And it's the same truth for adult as well. And we also find other studies have been shown early colonization of Mutans Streptococci actually indicates a much higher risk for children to develop caries later in their life and also into the adult dentitions. And the current study in adults and some of our small clinical trial in children also show if you just put in fillings or just do oral hygiene care or just fluoride prevention, those bacterial levels will not stop. And as we see as clinicians, for some of the cases, Dr. Tuan show S-ECC children who are going to be one of my population studied in here. Those children who go through extensive treatment under general anesthesia, and we do extensive prevention as we could in the United States. As much as that doing is not only at UCSF, 70% of them coming back with new cavities. So my questions as understand, I know diet plays an important role, but what the microbiology play. And what I want to understand is also we see that those children or adults who don't have caries, usually still 80% of them could have mutant streptococci in their flora, but at very low level. And what are the factors make mutant streptococci overpopulate and to start to induce decay? And we believe those key virulence factors are genetically determined. So we take that study approach, trying to identify potential virulence genes by comparison full genome sequences of mutant streptococci isolates that we isolated from age and gender matched children with severe early childhood caries decay, like Dr. Tuan presented. They all have more than 10 decayed teeth in their mouths compared to those who don't have caries at all. So the study was funded, I believe, started in 2006. So it's 8 years ago when I was funded with one of the Can-Do pilot study that we actually isolated mutant streptococci from 20 children with severe early childhood caries, and with helps of our pediatric department, because it was the hardest to find 20 age and gender matched caries-free children, even in San Francisco. So we managed to find them, and we isolated and enumerated mutant streptococci from the saliva sample. It's actually a swab, gingiva swab above on the tooth surface. And we genotyped them using what we call AP-PCR. It's like a fingerprinting. Usually if a child is not genetically correlated with another person, it's unlikely the bacteria they got will be the same as another person. But we do see if they matched, and we see in one of Dr. Boyd's population, I touched on to do a transmission study. They do happen in kindergarten after 8 months, that they can transmit to each other. So we did fingerprinting, isolated those bacteria, and we studied a number of virulence factors, but I'm only going to present 2 of them, which include acid production and acid tolerance. And we also studied another factor we believe is very important. It's called mutation formation. Mutation is actually a small peptide protein that produced by Mutans Streptococci as an antimicrobial molecule. And they actually help them to compete with the other good oral microfloras so they can overpopulate. And based on our DNA fingerprinting, we find it out. For the top, you can see the typical patterns for children with severe ECC. You can see usually if the patterns are the same, they are the same strain. So for the children who have severe early childhood caries, they should usually colonize with more than 1 to up to 5 different strains. Whether those children who are caries-free, they usually only colonize with 1 type of mutant streptococci, and we still need to find out how those different strains interact with each other. And when we studied the mutation, the specific antimicrobial peptide that mutant streptococci produce against other itself and cell groups and other good bacteria called sanguineous. And Streptococcus sanguineus is one of the housekeeping microflora. If it's high in quantity in the mouse, then you're going to be caries-free and also against the Bacterial disease. So it's actually the good flora help us to stay caries or periodontal disease-free. And usually it has much stronger portion to hold on compared to all those carcinogenic or the periodontal pathogens. And only when specific circumstance or when the very virulent bacteria come in, they can knock sanguineous down. So when we compare those 2 groups, and the caries-free is in blue and the caries-active is the ECC children is in red, we find that mutant streptococci isolated from children with severe early childhood caries actually produce more mutation, those antibiotics, to help them compete with other healthy oral flora. And we also find that mutant streptococci from children with severe early childhood caries actually also are more acid tolerant and produce much more acid at very low pH. That means they actually make the tooth dissolve faster and more extensive. And we studied all that, so we come with the conclusion, no, we know those bacteria are more virulent. But old assays, when I did those were traditional microbiology assays. And I was like stuck there. I know something is there, but what could I use this for the clinic? How could we use it to identify those virulence factors and also to use it for the future prevention in the clinic or at a population level to see, we see high-risk population here, what should we do? Then that's the time when I actually found Dr. Joe DeRisi, who is a microbiologist actually from the School of Medicine, and he introduced me to Dr. Fishbach and Mohamed, and we started a project of full genome sequencing, which Dr. DeRisi developed the Illumina high-throughput sequencing system, bring down the cost from about $20,000 a string to about $50 per string. That enabled me. We don't have a lot of funding to do this project, so I'm going to hand over to Dr. Donia.
Thanks, Ling. Yeah, I'd like to echo Ling in thanking the organizers again for allowing us to speak here. And yeah, this is a great opportunity, and it's really great to be able to get the other point of view. I'm a truly basic scientist, and it was really good for me illuminating to understand the other point of view from public health and all that. So I'm going to first start by stealing two quotes from this morning's talks. The first one by Dr. Saskia, when she said that integration and multidisciplinarity is an important part of our work, which it definitely is, but I would like to just bring into your attention that integrating basic science also will help. And I hope by this talk that I will be able to convey this message. The other point that I will quote as well, and that was from Dr. Habib's talk, when he said, nothing is as simple as sugar gives you cavity. And that's totally true. And hopefully by complicating things a little bit more in the next few slides, you will understand that, yes, it's not that simple. So in our lab, what we study is the human microbiome, which was mentioned a couple of times today. And we say human microbiome, but what does that really mean? It really means that any microorganism that lives on you or in you at a certain time, we consider that as part of the human microbiome. And this could involve good bacteria or bad bacteria. And just key figures here that are really like tragic figures is that there's 10 times more bacteria on our body or in our body, more bacterial cells than human cells. So if anything, we're more bacterial than humans, which is crazy to think about. And then, there's 100 times more bacterial genes than human genes. So that's another point. The other point is that, the human microbiome is very rich and very diverse. And it differs from person to person. So if we take 10 people from this audience and we examine their human microbiota and different body sites, we will find that there's a huge amount of diversity. And this diversity has been connected to disease and to certain types of diseases such as obesity and diabetes, where only fixing the microbiota has given a good outcome, a good therapeutic outcome, which is also another crazy thing to think about. But in our lab, what we're really studying is how do these microbes, tens of thousands of bacteria that live on us or in us, how do they actually interact with us and with other microbes on us? And how does that affect our health? So today I'm just going to give you a quick run through one aspect of this interaction. What is the most common language that all bacteria use to communicate with each other and with their host, us? And that is small molecules. So bacteria can use small molecules to do many, many things. For example, they can use them to scavenge trace elements. They can use them to fight each other. They can use them to work together, collaborate together. They can use them to evade the immune system so we don't see them and we don't kill them. And how do they, before I move on to the oral part, which I know you're interested to hear, how do they make small molecules? Bacteria are very efficient in making small molecules. They encode a certain number of genes in the same part of the genome. So in this case, I'm showing one example, 7 genes. They can encode 7 genes in the same part of the genome. These genes will encode enzymes. These enzymes will work together to form a final molecule. So this is a molecule that's made by bacteria, and that's how they make it. It's very efficient and it's very simple. This also allowed us to know what certain genes are involved in making these small molecules, and allowed us to, just by looking at the genome of these organisms, predict what kind of molecules will they make. And not only that, but what is the structure going to be, and what is the possible activity going to be. So what is the relationship between that and the oral health? We found that Streptococcus mutans, there was this paper that was published right at the time I joined my postdoc, is that Streptococcus mutans actually makes a small molecule that's encoded by these types of genes that I just told you about, and no one knows what this small molecule does. So we thought, okay, this could be one type of interaction between these bad, really pathogenic bacteria that cause caries and us or other good bacteria that live on the tooth surface. But the very cool thing was that it's not every isolate of Streptococcus mutans will make this molecule or will even have the genes to make this molecule. But some of them will lack these genes completely, and some of them will actually have other genes that will make other small molecules. So we partnered with Ling, and we started asking the question, is this variability in the gene content of small molecule making, is this variability correlated to caries at all? So our approach was to take 80 clinical isolates from Ling that were isolated either from early childhood caries or caries-free kids, children, and then take a multidisciplinary approach, sequence all of them using Illumina, isolate the compounds that they're making using a lot of chemistry, and then doing some functional assays to understand what these molecules, what the activity of these molecules is. So I'm just going to give you the end of this, which we found 15 types of genes that are all involved in making small molecules. And these could be present or absent from each one of the isolate. So here in rows, you can see the isolates. So SM3 is one isolate, SM22 is another. And these are the types of genes that we found, mostly new genes that we discovered. And you can tell immediately that each strain can have 1, 2, 3, or even up to 4 of these genes. So is this correlated with its virulence? Is this correlated with its competition capacity with the good bacteria that live on the tooth surface? We hypothesized that that is the case. And you can test that by just putting them on plates against each other. So here I'm showing 2 of the Streptococcus mutans isolates, which have different patterns of these genes that we thought make small molecules. And you can see that one of them was able to kill sanguinus, which Ling just told you is the good player in the game here, the one that actually protects us from other bacteria coming in, protects the tooth surface. And another one couldn't kill it. So does that mean that this one will be super cariogenic and this one will not be able to cause cavities? That is the question we ask next. But before testing that, again, in a model, we went after the molecules themselves and we tried to characterize them chemically. And I'll just give you one example of that, of how we do that. We actually take a genetic approach, pure micromolecular biology approach. We knock out the genes that are involved in making these small molecules from the genome of the bacteria. And then we grow them and we extract them. And we use HPLC, liquid chromatography, and mass spec to find peaks that are missing from the knockout but are present in the mutant, present in the wild type. So you can see here these traces are virtually identical except for this peak that now is missing after we knocked out the genes that are involved in making the small molecule. And we can isolate the compounds. This is a new antibiotic that we found being made by Streptococcus mutans. And we can show that it actually does kill. This is the wild type. If it has the genes, it kills. If it doesn't have the genes, it doesn't kill. But we wanted to use a more advanced model to test for this on actual teeth. And that's when Ling came in with this really cool model that she's going to tell you more about.
So I got those, the bacteria, the knockout bacteria that excited our Dean, Dr. John Featherstone, so much. And we put it in what we call it artificial caries model. So we basically use Bovantes, polish them, cut them up, and paint windows on them, and suspend them in the medium that mimic oral saliva and environment, and supplement them with different amount of sugar. And we basically had put either the knockout or the white type bacteria into the model. We incubate them for 8 days and change medium every 2 days. And after the end of the 8-day incubation, we take the bacteria out, wash them, take the biofilm, just cover where the open window is, and measure the bacteria species the numbers, and also measure the calcium loss from the enamel block, which means it's actually equivalent to the lesion depths. We actually also have done micro-CT data, and it's actually just finishing today, and I'm getting the result on Friday. So actually, then we also have the monoculture groups on the top, just as a control to see how those bacteria affect. And the two co-culture group, we co-culture Streptococcus sanguinis with the white type or Streptococcus mutans with knockout type to see whether the bacteria with or without the gene could compete well with sanguinis or not. And if you look at this graph, shows the Streptococcus and mutant Streptococci level presented in logarithmic values because there are usually logarithmic growth. And you can see the red is the white type mutant Streptococci in the biofilm. In the oral cavity would be dental plaque. And also the blue is the knockout strain. The numbers of mutant Streptococci in red and blue is comparative. And the condition we give them is a very acidic condition, means that's a lot of sugar diet that the child is taking. And we can see mutant Streptococci is comparable in the co-culture in both of the group, no matter it's knockout or white type. The only thing with the significant difference is the white type strain with the specific gene NN2 gene, that's the new antibiotic Mohamed group detected. With that gene, mutant Streptococci actually inhibit the colonization of Streptococcus sanguinis. It's about a 2 log difference. That means 100 times less than the knockout group when we just take up that gene. And if we look at all 5 groups with the bacteria, with the calcium concentration, that's actually the decay that was produced by this the biofilm, and we see the monoculture of mutant Streptococci alone makes the most significant amount of the decay, and Streptococcus sanguinis monoculture alone makes the least. We're followed by the white type co-cultured with sanguinis. We see there's only very minimal of sanguinis in this co-culture, and it's actually statistically significant, different from the either the monoculture of mutant Streptococci and also the co-culture of knockout strain with Streptococcus sanguinis. And actually, the co-culture of the knockout with sanguinis, it's not different from Streptococcus sanguinis alone. And what this in vitro model tells us that novel detection of the NN2 gene products in mutant Streptococci mutants did not affect the carcinogenicity of mutant Streptococci alone in the monoculture model, but it plays a significant role in Caries formation when it's co-cultured with the good health bacteria Streptococcus sanguinis. And the mechanism is actually basically facilitating mutant Streptococci to compete against Streptococcus sanguinis. And so in this small cohort of the samples we studied, as Mohamed mentioned, we actually identified 15 genes, including 2 subspecies of mutant Streptococci, 9 in Streptococci mutants, and you can see in the genome it's randomly actually assigned in any of the location. And we hope to take this to the next step is we want to learn more about how those molecules interact with other bacteria. Some of the molecules, like NN2, might play as a bad player, and some of the genes might play as a good player. They actually cooperate with the good bacteria, and some of them may not have a significant effects. And also we want also look at back to the population, because we have very small population here with 20 children in each group, to see if the presence of this gene actually leading to future development of dental decay or actually correlates with the dental caries. In that way, once we identify a specific gene that's linked to good or bad causing tooth decay, then we can use it using molecular techniques or child-side techniques that we can develop to screen those presence of those genes. So also maybe knowing the mechanism, develop specific regimens that antibiotics we can treat our children so we can stop decay or prevent decay before it happens. And I want to take that opportunity again to thank for all the group, my collaborators, including OLAB, on the very right side, Michael Fishbaugh and Mohamed's group in School of Pharmacy, and Dr. Darisi Lab in School of Medicine to allow this project to be conducted. Thank you.